So hello, everyone, and thank you for joining us for the final episode of this season's Data for the People presented by RockyDU Science Outreach. My name is Devin Collins, and I'm Program Manager for Outreach and Engagement at RockyDU. Data for the People is an interactive web series that invites all people to engage with relevant scientific research topics presented by scientists from our communities. The theme for this season of Data for the People has been people and medicine. And during the season, we've explored the science of how we study ourselves. We've explored our complexity as organisms, how our identities and characteristics can create challenges and opportunities for understanding humans and human health. And we've also looked at things like how historical biases and power structures have influenced scientific knowledge and continue to influence scientific knowledge and biomedical studies. We've also thought about how we've been able to get better and how we can continue to get better at studying ourselves by creating better, more comprehensive, and more just studies. So I am very excited today because we have many of the speakers from this season here to answer more of your questions and lend us more of their thoughts about this season's theme. So the RockyDU team and I have come up with a couple of questions that we can kind of spike into you all. But really, we just want to open this up for folks in the audience to ask more questions about people and medicine. I'll remind everyone that we are not giving any sort of medical advice. We are not medical doctors. We are all PhDs or students and postdocs. And so we are not qualified, or neither would it be appropriate for us to give you medical advice. But we can think a lot about how scientists like us can study people. So please try to keep your questions tailored to that theme. So why don't I give everyone just a quick chance to introduce themselves again, just tell us who you are, what you like to study, and what organization you're working with. And I'm gonna start to my left. The first person on my screen is Jeannie.
Hi. I think my dogs might start barking, so I apologize. But I'm Jeannie Garbarino. I work on the RockyDU team, and I am just really excited to be here for another season of D4P. So this is my third season working on this project, and really glad that you all could tune in.
All right, next on my screen is Victor.
Hi, everyone. I'm Victor. I'm a PhD student at Weill Cornell Medicine. And I really love to study the really integral mechanisms and inner workings of molecules and just really small things that relate to our biology.
Thanks. Lee, you're next.
Hi, Lee Bax. I'm a scientist at the Tri-Institutional Therapeutics Discovery Institute. We're a drug accelerator that works with Weill Cornell, Rockefeller University, and Memorial Sloan Kettering professors in preclinical drug development. As you might guess, I'm interested in translational research and taking steps that bring us closer to affecting patients' lives. I'm also passionate about mentoring people in their scientific career paths and introducing people to careers outside of academia. Glad to be here.
Thanks, Lee. Luth, you're next.
Hi, I'm Luth Hubiere, and I am a postdoc at MSK in the Alex Kansas lab. And basically my work consists of studying brain development and DNA damage produced by a protein called PBD5. And yeah.
Thanks. Angela.
Hi, I'm Angela. I'm also a PhD student at Weill Cornell Medicine, and I'm really interested in studying host microbe interactions. Right now I'm studying tuberculosis.
Thank you. Jude.
Hey everyone, I'm a third year grad student at Weill Cornell, and I'm studying the interplay between transcription factors and the initiation of prostate cancer.
Thanks, Jude. And then last but not least, Nora. Oh no, it looks like Nora might be frozen. Yeah. Okay. We'll give Nora a chance to introduce herself in just a bit once she figures out her connection. But in the meantime, I'm gonna jump right into my first question for all of you. And as someone who had the honor of sitting and talking to you at many different stages of like preparing for your talks and like getting to go to all of your talks, my big question for you is, and you can yell at me if it's too big, but you all covered a few really large and sort of overarching issues that really touch on almost every aspect of scientific research that in general, but also like especially scientific research that bears on humans and human health. So I'm wondering, like, for you personally in your field, what are some of the big questions or priorities that loom large for you? That you'd like to see us get better at, or see more data around, or whatever pops into your mind. And I think anyone can take them. Don't everyone jump at the same time.
I guess I can start. It is a really big question. And like I said, I study like the really integral mechanisms of molecules, but particularly, like I said, I study molecules that are super relevant to human biology. And, you know, like I'm sure many of us went over in our talks, those are mostly proteins. So of course, proteins are, running kind of a lot of the biochemistry and a lot of the mechanisms in our bodies. And I think what I'm kind of looking forward to in my field really is improving the techniques that we actually use to study the really core, integral chemistry and physics of like what's going on with the different molecules, that underpin a lot of our biology. I think there are a lot of like, in my field in particular, there are a lot of like, theories and kind of like big ideas about how a lot of things work, but currently it's just really difficult to test a lot of them because the technology and the I guess like ability to assay those things just doesn't really exist or isn't at the frontier of our like theory quite yet. So I think what I'm really looking forward to is improvement in those technologies, our ability to actually like assay certain hypotheses and theories.
Yeah, I think that's such a big job, right? I know in my own research, I studied addiction as a grad student and a postdoc, and it can be, so it's a very not fundamentally, but it is a very human issue that I would study. And we used a lot of mouse and cellular assays, but we can't ask mice how they feel, and we can't ask cells how they feel, especially. We can only kind of like, model the things that are going on in humans and make a lot of assumptions about what correlates to what, but actually being able to assay the thing that we care about and get at it without the complications of the way that culture or other aspects can influence what we think about people's answers or what their answers will be, much less what we think about the disease that we study. It's really hard. So really getting at the core of what you're and being able to assay exactly the thing that you need is a really tough order. So I'm wishing you luck. I hope it works out. Does anybody else want to jump in? I can also, maybe we can narrow the question a little bit to what is the question that you're most interested in that you think your work addresses?
I would just like to expand on what you guys, Devin and Victor, were talking about using mice models, because, and this is just more broadly in immunology, we use a lot of mice models, but mice have different immunity compared to humans. For one thing, they have short lifespans, and you have many more litters versus humans only have maybe a few children at a time, and we have much longer lifespans. So how our immune system reacts to different pathogens are going to be different than what mice are going to do. And so it has been difficult. And I think with COVID, there was this kind of people saw that there was this problem, that we know a lot about how mice work, but we don't know that much about how the human immune system works. And there is some technologies coming out where we use human organoids to try to more mimic the human immunology, but it's still not a perfect system, and it's still not in vivo. And of course, we can't really do a lot of in vivo work because it would be unethical for the reasons that we've discussed previously in the session. But yes, I think more technology for organoids and those type of systems might help.
Yeah, I think that's a really great point. I wonder, oh yeah, Lee, go ahead.
I just wanted to add to that from the perspective of drug development. I know that the fact that mice don't recapitulate human biology is a huge issue, because most preclinical work going into very early drug development is done in mice, and mice have been cured of cancer many, many, many times, but we don't at this time have very many ways to cure various human cancers. So, that's just a testament to the fact that mice are a challenge. There's lots of preclinical work done in other species, dogs, primates, other large animals, I think specifically for that reason. But, because of ethical reasons, obviously you can't go into a human until you have something safe. So there is also, I think, a push for these more translational models, organoid models, co-culture models, these like organs on a chip. There's a lot of really interesting like 3D printing and kind of 3D structured, model systems that people are trying to develop. But, as far as I know, a lot of that hasn't really been pushed through any preclinical development to the point that it was able to show translation in the clinic, because most of these technologies are new. But it's definitely an issue in translational science as well.
I think an issue that all of these are really excellent points of like where we should be trying to go, as an enterprise. But there's something that I think we haven't really discussed in this series, but I think it's worth mentioning here, in that if we consider scientific research to be sort of this interconnected body of knowledge, and it really is around the point of scientific research is to both generate and organize that knowledge into some narrative that's testable, and has logical conclusions and things like that. But there's so much science going on, and not all of it is as interconnected as it could be. And so I think there is a really big need for better information architecture when it comes to the output of scientific research. And that could be considered through the lens of like publication. So we've been talking about peer-reviewed publication. And there's other ways that people are starting to think about dissemination of scientific research. And where can we find ways to accelerate that and make sure that researchers can connect with each other to sort of start thinking about what those big picture pieces are. So I do think that there's this other layer outside of research proper that really needs to be solved as we continue to generate such huge data sets also. I mean, they're just ginormous, right? So those are just some of the thoughts that I had as a non-active researcher.
Related to that, Jeannie, actually a lot of, we have a lot of companies who come to us with various computational, things they're working on. One of the things I see a lot is companies who are trying to synthesize data from many different sources for the use of companies working in preclinical drug development to learn as much as they can about a target. I mean, if we have a new target, we go to a number of different databases that have, big data, whether those are like the Open Targets platform, which is great, OMIM for phenotypic data in humans, JAX for phenotypic data in mice, Protein Atlas, Cell Atlas. The Broad has like a bunch of different resources. Like there's companies who are trying to work to bring all of this together so that you can look for a target and they can give you all the papers where that particular protein or gene was mentioned, all the patents where it was mentioned, all of the websites that mention it, all the assays that involve it. I think that's a really kind of a good step in that direction. But what's unfortunate is that a lot of pharma companies who have these types of things, they're like proprietary. So only they have access to it. So I think, you know, a push for an open source resource like what's provided by Cell Atlas, Protein Atlas, the Broad, you know, open source software packages or, you know, computer programs that allow you to kind of synthesize all that data in one place when you're interested in one particular target or pathway. It's a really important thing that needs to be pushed.
Yeah, I think pushes for more open data and more, or especially more open source tools that we can use to compile all of the data that's out there and bring it together in a way that makes sense is so useful. And I know in my own practice, I was able to make use of things like that, like some of the openly available things that like Jackson and some of the things that are on like library, you know, National Library of Medicine sites and all of that. I'm wondering, how have you all been able to use those sorts of tools in your own research? And maybe if you have a favorite that you can let us know.
Well, for me as a neuroscientist, I have one of my favorites is the Brain Atlas from the Allen Brain Atlas. And thanks to that, there is like a huge resource on like how on development of the brain and the full developed brain, and just not like an atlas for the mouse brain with all the parts that you can always use as a reference, which is very important because it's very complicated. But also they are like having a huge effort also developing now the new single cell technologies and generating maps, like reference maps. And that I think is very important for us researchers to have that and be able to use those tools to then study diseased brains and have this reference map for the study for the brain.
One tool that I've used is QIAGEN's Ingenuity Pathway Analysis. And so this is, it's a company, but you can put your RNA sequencing data into this database. And so when you get RNA sequencing, you have like, 20,000 genes, could be more, about. And so if you were to individually look at these genes, it would be really difficult to analyze each gene. So what you can do is look at this gene set enrichment. So you have like different pathways, and then they can tell you what those pathways are. So you're looking at within a pathway maybe, and also because RNA is very transient. So a gene might be upregulated at one time and then downregulated if you don't have the right time point. So if you're looking at multiple genes within a pathway and like 7 out of 10 of them are upregulated, that means that probably this pathway is upregulated or being expressed. And so it's really useful to get back that data of what pathways are being expressed.
Yeah, I really love that idea of being able to see like along different points in maybe some sort of I'm trying to think of a good way to explain, but like seeing like along the course of a biological process, like what sorts of endpoints you might see, because sometimes we only get to see a snapshot of what the biology really looks like at one given time or in one set of conditions, and being able to see like, oh, this gene is upregulated here or downregulated here is really cool, because we get more of that global view and not just in one particular way. And that's so important, and I don't know if a lot of scientists like always think about it that way. I know for me, like I would get really fixated on one particular experiment and the outcomes there, but not think about everything. And, you know, studying the brain like I did, like you really have to think about a lot of different things at one time. Not that the brain is special, but I mean, it is special. I think it's special. But, you know, in other organ systems, it works really similarly. Everything is so complex. Yeah, so I think those are like really great tools. Are there any other tools that you're sort of interested in? I'm thinking about, you know, like big initiatives like the Human Genome Project or like the Brain Initiative, are there other big, like maybe government funded or institution funded or international collaborations that you have benefited from, or that you would like to see, or around a specific kind of set of tools, or delineating a certain set of biological processes?
I think probably the two most invaluable for me as kind of someone that works in like molecular and like structural biology are first the like huge like NIH database of like genes, like RNA and protein, because there they kind of outline pretty much all genes that that we know of, and they also catalog like various like human genomes that have been mapped, and they on those genomes they map out like all the like translated proteins, and also the papers that are like associated with all those genes and translated proteins. So it's like super invaluable just to like be able to look up any gene that you want and get all the information on it really quickly. And then the other one is the protein data bank, which is especially really useful for structural biology, because there you kind of get a catalog of all of the various protein structures that have been resolved experimentally. And I think also now that like AlphaFold is a big thing, they also catalog protein structures that have been predicted as well. So both of those are super, super invaluable for molecular and structural biology in particular.
Yeah, I think tools like oh, Luth, you go first.
Sorry, now that you touched, Victor, the AlphaFold thing, I think it's very powerful, because I was just like commenting that with my coworkers, that they are also developing this new algorithm where they simulate also not only the protein folding, but the protein folding together with DNA or other structures. So this is very powerful. I mean, it's never a definitive structure, but it can give you an idea of like when you don't have a crystallized structure for the molecule you're working with, on how it's interacting, just not by itself, but with other partners, other proteins, complex. And I think it will be very powerful for the future of predicting these kind of things when you don't have the actual crystal structure that takes years to get, right? So Victor, you sure know much more about this than me, but the part of the lab that is interested on that, they were like very excited.
Yeah, these days they actually can also simulate things like different like drugs binding to the proteins and stuff, and they can predict like what the structure is going to look like with the drug bound or like various other molecules. So yeah, there's a lot of strides in the field of like computational prediction of molecular interactions and stuff. So it's really exciting.
Those resources are actually really heavily used in the drug development industry as well. There's a lot of people putting efforts into like AI driven and other computational methods for driving drug discovery. So if you have a really high resolution structure of a compound, that can be used to run virtual screens and other ways of looking for potential binders. You can't yet look at it and say, this is the compound that'll fit in there. Like, we're not there yet, but I hope someday.
I think
So, go ahead.
Yeah, I just a comment. I just think that these are all so, it's really amazing. Like thinking back to the comment that you made, Lee, about the necessity for a lot of these open source projects and databases being associated with one another and working in tandem. I really love that we can, we're at a point where computationally we can take a lot of this data, make it into meaningful representations of the biology, and then see how all those different once disparate databases and structures really work together to get a better model of what actually is going on, and not just having to imagine or having to think about like, well, maybe if I look at this time, or maybe if I catch this event here, like you get a better idea of when and where to look in cells, or when and where to look in whole animals or whole people. And that's a really amazing tool that opens up a lot of biology and a lot of different kinds of ways of thinking about our experiments for us.
So there is a question from the audience, and this is really interesting because they're bringing up folding at home, which we know is taking place at Weill Cornell. So On the topic of open source tooling and data collection, is anyone here familiar with Folding at Home? We are familiar with it, or at least some of us are, and similar projects. How helpful have they been in progressing research?
This is so crazy because I use Folding at Home actually for my research. So it's kind of cool to see that, you know, people actually know what it is. I thought like nobody knew what Folding at Home is, but it's like absolutely integral to my research because I actually use it basically to study, or rather to predict computationally how like various molecules are like interacting with others. And it's been like I said, invaluable to me because it basically enables you to do various computations that would be like impossible to do with just like single computers or just like even like a computational cluster. And for anyone that doesn't know what Folding at Home is, it's basically like a distributed computing network, which basically lets you like borrow the computing power of like volunteered computers to make like one giant supercomputer. And I think at some point last year, 2 years ago or something, it was actually the like most powerful supercomputer in the world or something. So cool to see it mentioned here.
Yeah, we had one of the developers of Folding at Home join us for a summit that RockyDU hosted a few weeks ago, and it was really cool to hear the stat that they accidentally built the world's largest supercomputer for like several moments in time or something like that.
Yeah, that was a really cool piece of trivia that we got to learn. So I kind of want to change gears just for a moment, maybe for longer than a moment. But I'm thinking more about, you know, again, like the theme has been people and medicine. And we've talked a lot, you know, we've just gotten done talking about a lot of the tools that we use or the tools we'd like to see. But I kind of want to think about the people. And I know, you know, for example, we heard from Nora and Lee about how we can make more just studies that really honor the communities that we are working with, and understanding, you know, how to do things like build trust. And I wonder for all of you, but maybe specifically starting with Leonora, what are your thoughts on how we can be better at you know, ethically constructing studies that involve human participants?
Yeah, sure, I can speak to that. I'm Nora, by the way. I didn't get to introduce myself earlier because I was having some technical difficulties, but I work at the Tri-Institutional Therapeutics Discovery Institute along with Lee. So I guess I'll kind of summarize our talk because we talked a lot about this, specifically focusing on Indigenous communities and people doing research with Indigenous communities, particularly genomics research. And sort of a lot of what we talked about was how this could be done ethically. And basically it kind of boiled down to that researchers and the communities should really work together in partnership in order to do these studies, and should really create sort of close partnerships and trust and build trust that the data is going to be used ethically and with the best interest of the communities in mind. And so that's sort of, yeah, referred to as like community engagement. And I think that that's a really important aspect of doing research on humans, is not just treating them as subjects, but as treating them as like you're giving your DNA or whatever you're giving to this project, and that deserves credit and respect, and should be treated as such, and should in the end help the communities that are being researched and not do harm to them. So yeah, that was sort of the thesis of our talk.
Yeah, I think that's such a good point. As I was asking you the question, I felt myself having to stop and say, I've gotten so used, just because I've heard this so much, human subjects, human subjects, but really it's participants, or maybe that's not even the best word. That's the word I like to use right now. But you're taking that, I think, even a step forward, and especially, some of the themes that you covered and the actual initiatives you covered in your talk was really human partners, like research partners. It's not just that people are participating in research studies or should participate in research studies. They are making it possible to do the work in the first place. And so it really is a partnership. And, those partners might be contributing different things. Like some of them may be contributing, offering their own like biological material, but that doesn't make them any less of a partner in the research. And I think we have to honor that and think about that from the bottom up, not just bringing that in after we decided, oh, we need this from X group or X people. Like really thinking about just the idea that it is through the generosity of folks who interact with us that we're able to get done the studies that we want to.
Maybe I'll just add on to that as well. Like another theme was sort of thinking about the impact that research has, and particularly for groups that have been historically taken advantage of for science and other things. Like, there should be particular care taken there to sort of gain the trust and respect of those groups, and make sure that the outcomes of those research are like culturally sensitive and actually helping the communities in the end.
I wonder if anybody has thoughts on where, like we know that scientists, and I think we talked a little bit about that in many of the talks, like scientists have specific sorts of responsibilities to the people who participate in their studies, or just to, just ethically, there are certain ethical obligations that we have in general, no matter what kinds of science we're doing. But I wonder where else, the responsibility might lie. Is it do you see like a role for policy or governments and kind of directing what scientists can and can't do? Like obviously there already are many policies and sorts of rules in place, and funding agencies might have their own rules, but I wonder like what are some of the big things that or where might we like look for guidance or power to be able to institute rules that we need to keep ourselves honest or keep ourselves ethical?
So I think just talking about what rules are already in place, or what regulations or institutions are already in place. We have something called the IRB. I think it stands for Institutional Review Board. So this is a council of people within an institution that, in order to do any type of human subject research, you have to get approval from them. And the board has to consist of at least one scientist and one lay person, so someone who's not a scientist, to represent the general public. And they have to weigh the risks and benefits of the study, and to make sure that the benefits of the study outweigh any risk, and to make sure that there is no undue harm onto the participants. And yes, and you have to go through this process anytime you want to do human research. And with that said, like there are criteria for what is considered to be human research. So if you are doing something completely anonymously, you don't know where let's say you receive samples from somebody and you have no idea where those samples came from or who the people are, that would be considered not human research because you don't personally know who those people are. But you still have to get IRB approval to make sure that that is considered not human research. So that is something that's already in place.
It might be worth mentioning that while we do have this institutional review board in place, which is an essential component for people to be able to provide their informed consent to be in participating in research. I think there's also issues with who actually is being consented and who is being recruited. Like, we're not recruiting a representative sampling of our population in the United States for clinical studies, right? And because there isn't representation in clinical studies, there isn't necessarily representation in how we develop medicines and treatments and drugs, right? And that might be actually a problem. So I think a question that I've always had was something around, like, how can we improve the relationship that science has with communities? And I know we sort of talked about it a little bit through the paper discussed by Leah and Nora, but there's got to be better ways to sort of think about community engagement, participatory science. Like, for example, I know there's been a lot of progress through communication of science through barbershops as one example. And I know there's a lot of people working to do vaccine communication through faith organizations, right? So what is required for science to showcase that it is actually trustworthy, or at least a lot more trustworthy than it used to be?
I could add to that. I think one effort to wait, hold up. Am I echoing?
No,
You're good. We're good.
Yeah.
Oh, all right. Yeah, so I think one effort to meet that end is so before in clinics, like people, doctors or clinicians used to like stratify patients based off like their like definition of like their ancestry, right? But nowadays, like people are like using genetics, I would say, to define people's ancestries, and that better serves patient stratification and development of drugs, because like now like the studies that we do here in the lab would better like represent what's happening in a specific patient, rather than like using like a small subset which doesn't represent like all patients.
Yeah, I think it's interesting to be able to get like and especially in places like, I mean, I live and know, the US the best because that's where that's where I'm from, but thinking about places where there are really big admixed populations of many different people who may or may not share ancestry from many different places, it's really, I think, one really great thing that we can do better now, and that we can continue to do better, is to recruit a lot of folks irrespective of our own assumptions about where they come from or what their genetics or what their ancestry might be. We can actually just look and find out, like, okay, of the people who have volunteered to take part in the study, what are their biological characteristics or what are their genetic characteristics? And, they may report one thing that more aligns closely with their own culture or with their family history. But, if we see something else genetically, it doesn't contradict what they've said. It just adds to the richness of the information that we have. And we have better tools to be able to think about it in more complex ways now. Yeah.
I think there's also the other side of the equation, which is sometimes people are participating in the studies because they want to receive some monetary compensation. And so there may be certain populations that are more vulnerable to be doing riskier research because they're in an impoverished situation. And so that's also something important to think about. And I have to say, it is really difficult to get a diverse, to recruit a diverse population in my own research. And yeah, I'm trying to think as well of how to better get more diversity, but also without not coercing, but like without targeting people who just want it for the money.
Yeah, that's such a good point. And it's something I don't always think about, excuse me, myself, but really when we're thinking about partnerships, like one of the things that people should be compensated in some way, or they should be, there are things that we need to do to take care of folks. So I know for my own research, we would do, some inpatient or outpatient care for people who are people with addictive diseases while they were in our studies. And one of the benefits for them is that they got to, if they didn't have insurance, or if they were, for some other reason were not able to access medical care, they could at least get that care at the time that they were, in the hospital doing our, working with us on our studies. But then that could also, things like that can also bias the populations that you get, because maybe folks who have other routes of care for the same disease may not participate in your study because they don't need that care, or they don't need the money that people can offer, or things like that. And it's really interesting. I know, at least in my field, there are folks who, who are studying that and understanding like why, trying to understand why folks are making the decisions they do, not only about, the care and management of their addictive disease, but also, how they participate in studies. It's interesting to think about that, but I love that there are scientists who are looking at those questions. So I think I would love to hear this question has sort of been rattling around in my head for a couple of weeks, ever since your talk, Ruth. You talked a little bit about psychedelics. You talked a little bit about psychedelics, and one thing that I know I encountered in my own research is that you couldn't. It's tough for some scientists to study certain drugs or use certain kinds of tools because of social stigma, or even the laws that are on the books. I wonder if you have any thoughts about, what sorts of policy changes, or what sorts of, things we can do to fight stigma against, about drug use or other diseases, or, to be able to study them better. If there's anything that you have in mind.
It is very complicated because everything's tied up. It's not like science is separate from the population issues. And I was gonna make a comment before, but I guess my time passed. And on the topic before, like sometimes the government, like as I said in my talk, can forbid to study 13 things, right? So you still need these regulations in place. You need the regulation so psychedelics cannot be misused by people, but can be used as a way to just like treat patients, and just not psychedelic opioids or any other type of drug that right now it is useful in some ways for humans, but misuse can lead to disease and health problems. So it is very complicated for this reason that if you release the law, like lax the laws, and then people can use it broadly, you end up with a problem. But if you restrict it so much and stigmatize it so much that you decide this is not helpful at all, then you end up stopping the research progress. So there is this moral battle, right, between science and community, and I guess you made a point before that for us is to educate people, to educate for both the risks and the benefits. And I know I'm not from the US, but I have such a different relationship with how like view of drugs, because I came from Spain, and I know like when I came here that there is a lot of people that tells me there was this huge campaign against drugs that demonized it and demonized their use, versus back in Spain I never had this huge focus on that. Like I had some education on drugs, but I remember people coming and telling me the risk of taking the drugs, but the benefits of how to use them in clinic. And I never fear them, or it was just like a respect right? Like, and here, when you over stigmatize something, you can get the opposite effect. So you end up like having people that ends up very, like you want to break the rules, and then you end up abusing the drugs, versus like if you understand, look, there is a risk in everything that I do, and I don't want to get into this field of like, I don't know, taking psychedelics, because then I know what it can do to my brain. So you understand, and then I don't know, it just, for me, it is this balance in between giving the information and then trusting that people is going to make the right choices. And you're a drug researcher, and I believe like in the touch of drug addiction, especially, people usually don't get addicted to drugs just because, like, usually they get addicted to drugs because they are using it as escape. So people that they don't usually have like situations that are like ideal in their environment, like, and obviously it's a very complex problem. But anyway, I think putting these models is good and bad in science, and as a scientist, we have to educate the people and believe that they make the right choice, and also the government should do their part, where like they take care of their people, and then you end up with less people in bad situations that they end up, abusing this type of substances in that case. And then obviously there is a lot of other like stem cells and other people that don't believe, there are a lot of debates and moral debates that you can get into the weeds into those. But yeah, I think the solution as always is education and trusting that people with the right knowledge is gonna make the right choices in the right space. Like, obviously I'm not saying, but yeah.
Yeah, I love that you bring up education and like giving people just like access to information. I guess, that's what D4P is all about, is making sure that everyone is able to access the information that we can, I think as scientists, can kind of take for granted that access. I have one final question for everyone. It will hopefully be a little bit more fun. Not that these have not been fun, but I want to have like a definitely fun question for you. And that question is, we have like, we've all been talking about humans and health for this season, and I would love to hear from all of you what excites you, or what do you love about studying humans and human diseases? I can break the ice too if you want. The thing I really liked when I was at the bench, the thing that I really liked was that I got to, all of the labs I ever worked in had to do with human disease and health, and were translational. So the things that we studied in the lab had direct bearing on, actual illnesses that people have and experience. And it was really cool because I got to meet a lot of the folks who took part in our research. And especially when I've studied things like rare diseases, like rare neurological diseases, we got to know the families that like were carriers of the disease mutations that we looked at. And it was just really cool to be able to, again, like have that partnership with them, and we shared data with them, and they helped us to get funding, and they were able to like come and tell us very personally about the symptoms of their illnesses and really describe in detail. And we were able to have like a personal relationship. And so being able to see directly the like results, not even if we like couldn't find the quote unquote cure, we got closer with like things like treatments, but it was just cool to see how happy they were to work with us, and how much they came to trust us over the years. And that was really special to me. So getting to see the like impact, the human impact was really cool.
I guess for me, the coolest thing is just being able to bring, what I know from the science that I was trained in, which I was never trained formally in like biology or chemistry. I was trained formally in physics, actually. So for me, the coolest thing I think is just bringing, concepts from physics, which are obviously important to, biology and human health as like a larger topic, just bringing those concepts and, bridging multiple like interdisciplinary like sciences and disciplines, and doing all of that to kind of in some way, from my basic science research to improve, outcomes in health. I think that's just the most exciting thing for me.
I think for me, the most exciting thing is being able to better understand ourselves as humans, and specifically in like microbiology and immunology, it's something that we can't normally see on a daily basis. It's something we have to see under the microscope, and like seeing images of infected cells or seeing how our cells react is really cool. And then also getting results and figuring out better what more about ourselves that we don't normally see by the naked eye.
Okay, I'm gonna jump in for my answer to this. I think for me, I'm gonna be a little bit like really textbook nerdy and say that, so I did a lot of research on cholesterol, intracellular lipids, phospholipids, like triglycerides, things that make your membranes and things like that. So I feel really jazzed about intermolecular forces. Like I love thinking about how little tiny, so this is going really granular. I love thinking how little tiny changes in like a DNA sequence that leads to a little tiny change in the protein sequence, how that completely changes how a molecule can interact with its surroundings and with other molecules. So I love that kind of stuff. It's like a really cool puzzle. You don't even have to study whole body systems. You could just study a cell to figure that stuff out. You don't even have to study a whole cell. You could just study like, and what's inside your nucleus or something. It's like there's so that you can go so far through. I know intermolecular forces feel like a little boring in your textbook, but they're just literally the coolest thing in real life.
I can answer. I think so. I work now on drug development, making drugs and finding drugs to cure diseases. And I really like working on something that, can maybe really impact someone's life and be beneficial to people. And so that's very motivating, and that's the reason why I like working on drugs. Yeah.
I guess for me, the most motivating thing is to understand how our brain is formed and how it actually works, because it's one of the areas that is less understood in biology, because of the fact that we cannot take samples of the brain, because it's not like you can maybe take a sample of the liver and you still survive, but you cannot take a piece of your brain and do well. So I think for me that answering this question is the most exciting thing, and how, I work more in the development part, but I work with cellular diversity, so understanding how the brain is composed by different cells that are different and diverse to one another, and they communicate within each other is the coolest thing for me.
Oh yeah, I want to add to that too. So I think what's exciting in my research is so I like to think of cancer as like that toxic X that like never goes away. It kind of finds like ways to come back and like just make your life like, hell. And what I do in my lab is like to like identify like the key like transcriptional machinery and epigenetic machinery, which like transforms like normal prostate cancer to a normal prostate to prostate cancer. And that in itself is like super exciting, because if we can like actually like figure out like the key mechanisms which are being like disrupted, we can sort of avoid like the late stage, like really bad like forms of cancer.
Yeah, so getting better at detecting everything before it goes wrong. Yeah, those are all really wonderful answers. Thank you very